Hello student, good morning. Myself Sri K.D. Borse sir, biology subject teacher. Today we are going to learn 11 science biology subject syllabus contain chapter number 2, systematics of living organisms. In this chapter contains some concept study today, like concept chemotaxonomy, concept numerical taxonomy, concept phylogeny, and concept DNA barcoding. All these concept detail study in today's lecture. So let us start today's first concept, chemotaxonomy. What is chemotaxonomy? It is method of biological classification on the basis of similarity and differences in chemical constituents of an organism called as chemotaxonomy. Means chemotaxonomy is a method of biological classification in an organism contains some chemical constituents present or absent. On the basis of them, biological classification takes place. Those organism group contain same chemical constituents, they are similar groups. Those organism contain different chemical constituents, they are differences. So on the basis of similarity and differences in chemical constituents of an organism called as chemotaxonomy. Example, archaea is one of the form or domain organism. Its cell wall absent peptidoglycan chemical. Means archaea is one of the organism group, its cell wall not seen peptidoglycan. So this chemical constituents is not seen in an organism present in their cells, so called them archaea. Bacteria is also another form or domain. Its cell wall contain present peptidoglycan. Means this chemical nature present or absent different two groups or domains or forms of organisms on the basis of chemical constituent, absent peptidoglycan, present peptidoglycan. Also example, eukarya is also one of the form domain. Plant including eukarya, its cell wall present cellulose, and eukarya fungi cell wall present chitinous. Means here noted all three different domains like archaea, bacteria, eukarya, different chemicals present in their cell wall. Means in an organism contain chemical constituents on the basis method of biological classification. So here noted all examples prove that chemical constituent of an organism are different from one another. So this type of classification called as chemotaxonomy. Means this classification is especially depend on chemical present in an organism. Now we learn today's second concept known as numerical taxonomy. What is numerical taxonomy? It is also a method of biological classification based on quantification of characters using numerical algorithms. Quantification means quantity of characters, and numerical algorithm using method of biological classification. Such classification called as numerical taxonomy, and such numerical taxonomy first proposed by Sokel and Smith in 1963, who proposed first time numerical taxonomy. This is one of the method of biological classification. Sokel and Smith proposed first numerical taxonomy in 1963. Now we learn today's next concept known as cladogram. What is cladogram? It is a diagram. Cladogram is a diagram of three domains of life. You know, three different domains of life: archaea, bacteria, eukarya. So these three domains of life diagram represent hypothetical relationship between organism and their ancestors. This diagram are called as cladogram, and this cladogram diagram is a typical branching pattern diagram. Seen here diagrammatic representation: archaea, bacteria, eukarya. All these three forms or domains of life seen here common ancestor. Archaea, bacteria, eukarya are three domains of life diagram represent hypothetical relationship between organism and their ancestors. Means this one is common ancestor and these three are a form of life or group of life or domains of life. So this diagram called as cladogram. Means simply what is cladogram? A diagram of three domains of life. Cladogram is a diagram of typical branching pattern. Which one is typical branching pattern? Cladogram is a typical branching pattern. Up to this we detail discuss here. Cladogram is a diagram of three domains of life. Now we learn term phylogeny. What is phylogeny? Evolutionary relationship between a group of organism called as phylogeny. Such phylogeny help to understand evolution. What is the main importance of phylogeny? To understand evolution. Best example of phylogeny or a very good example of phylogeny. Phylogenetic relationship are three domains of life. You know, in three domains of life means three form of life. First one is archaea, then bacteria, then eukarya. Such three domains of life propose Carl, whose also second example, five kingdom classification. In that five different kingdoms, number one Monera, then Protista, fungi, plantae, animalia. So such five groups are completely depend on evolution. So such two examples are most important three domains of life proposed by Carl, whose also five kingdom system proposed by R.H. Whitaker. These two best example, very good example, phylogenetic relationship. Up to this we learn phylogeny term or concept. Now we learn DNA barcoding. What is DNA barcoding? A new method for identification of species. Means with the help of DNA barcoding, identification of species based on DNA sequence from a teeny tissue sample of an organism called as DNA barcoding. On the basis of DNA sequence present in an organism, such DNA sequence identify the species, and this method called as barcoding means species identify on the basis of DNA sequence present in an organism. Such method is specially studied with the help of a teeny tissue sample removed from organism. It contains cells and cell contain DNA. Such DNA sequence on the basis of DNA sequence identify species. Such method called as DNA barcoding, and such DNA barcoding method include two steps. Number one, first collecting data barcode or DNA barcode data of known species, means those species are exist. Such known species DNA sequence collection, also matching the barcode sequence of unknown sample. Unknown, those species identify, such identify species DNA sequence and known species DNA sequence match one to another. If matching the DNA sequence, so identify the species. Such method is newly method on the basis of DNA sequence of an organism identify of species. What are the different applications of DNA barcoding? Number one, first, DNA barcoding helps study newly identified species, also help understand evolutionary relationship between living beings. DNA barcoding also protect endangered species, means those species extinct in future, called them endangered species. Such endangered species also protect by identification of its DNA barcoding. Next, number 4, preservation of natural resources with the help of DNA sequence. It is also one of the application of DNA barcoding. And identify disease vectors. You know vectors, some disease caused by pathogens, and such pathogens are transported by vectors. So identify disease vector is also a application of DNA barcoding. Also number 6, identification medicinal plant. Those medicinal plants show a special DNA sequence. So identification of medicinal plant, identify disease vector, preservation natural resources, protection endangered species, help understand evolutionary relationship between living organism, also help study newly identified species are different applications of DNA barcoding. Up to this, we detail discuss DNA barcoding and its application. Now, on the basis of today's lecture, some exercise questions noted here. Number 1, which one is typical branching pattern? Yes, cladogram is a typical branching pattern. Who was proposed 3 domains of life? Yes, Carl Wood proposed 3 domains of life. Method of biological classification based on chemical constituents called as, yes, chemotaxonomy is a method of biological classification based on chemical constituent present of an organism. Number 4, which type of kingdom system proposed by R.H. Whitaker? Yes, 5 kingdom system proposed by R.H. Whitaker. Number 5, help study newly identify species is a application. Yes, DNA barcoding, a diagram of 3 domains of life, represent a hypothetical relationship between organisms and their ancestor called as cladogram. Match in column A with column B, here noted column A, column B. Archaea cell wall contain absent peptidoglycan. Bacteria cell wall, yes, present peptidoglycan. Eukarya plant cell wall, yes, present cellulose. Eukarya fungi cell wall, yes, present chitinous, means archaea cell wall absent peptidoglycan. Bacteria cell wall present peptidoglycan. Eukarya plant cell wall present cellulose. Eukarya fungi cell wall present chitinous. Up to this today's lecture we detail discuss here chemotaxonomy, numerical taxonomy, cladogram, phylogeny, DNA barcoding, and some exercise question based on today's lecture. Hope that you understood everything. Thank you.